So how exactly does a single paper cut just heal up without a trace? It's something we completely take for granted every single day. But how do our bodies magically repair that everyday damage and regenerate lost tissue? Well, let's dive into this explainer and find out. To answer that, we're zooming way down into the microscopic world to tour the ultimate biological factory, animals' cell division. We're talking about the incredible, highly orchestrated process of mitosis. Section 1, The Power of Mitosis. To really wrap our heads around how we heal, we first have to grasp the sheer scale of the cellular turnover happening inside you right this very second. Just take a look at that number, 120. That's the lifespan in days of a single red blood cell. I mean, they work incredibly hard, but they do wear out. And because of that finite lifespan, constant replacement isn't just nice to have, it is absolutely vital for our survival. So the crucial takeaway here is that mitosis is essentially a biological cloning mechanism. It's what we call an equational cell division. That basically means one parent cell perfectly maintains its chromosome count as it divides, and that results in the formation of two genetically identical diploid daughter cells. It's really a chronological hero's journey, mapping out this highly regulated assembly line. We kick things off with interphase as the prep work, and then move right through the active stages of division: prophase, metaphase, anaphase, and telophase. Let's go ahead and walk the factory floor. Section 2, Interphase, the preparation. Okay, before the big dramatic split can actually happen, the cell has to gear up. Just picture the vibrant cytoplasm expanding as it gets ready for division. During interphase, the cell is growing larger, it stays highly metabolically active, and, this is key, it duplicates its DNA to create a perfect carbon copy of its genetic blueprints, forming identical sister chromatids. Now, in a human cell, that means duplicating all 46 chromosomes. It's a massive undertaking. The cell literally has to ensure that both of those future daughter cells end up with the exact same instruction manual, completely intact. Section 3, Prophase and Metaphase. Now we hit the action-packed main event. Watch as the magic starts in prophase. The genetic material condenses from this kind of loose jumble into visible, tightly packed X-shaped chromosomes. Then the protective nuclear envelope breaks down, and these incredible spindle fibers start reaching out from the cell's poles like microscopic cables. And then we hit metaphase. During this stage, the factory is in strict, perfect order. Those spindle fibers we just talked about, they've attached to the chromosomes and anchored them directly on the cellular equator, right in the middle of the cell. They line up absolutely perfectly, end to end. But wait, before anything separates, we hit a wall. This checkpoint is a crucial quality control stage. The biological factory will actually halt the entire assembly line until perfect bipolar tension is sensed across those spindle fibers. The cell is literally waiting for a molecular green light, confirming everything is perfectly aligned before it allows the division to proceed. Section 4, Anaphase and Telophase. Once the cell gets that green light, boom, the tension breaks. This is the peak excitement of our factory tour. Anaphase. The cohesin proteins that were holding those sister chromatids together break down instantly. The spindle fibers retract cleanly and rapidly, snapping the chromatids apart and pulling them to exact opposite ends of the elongating cell. Finally, the dust settles in telophase. The newly separated chromosomes arrive at the poles of the cell and begin to decondense, just kind of loosening back up. The factory then starts rebuilding its nuclear administrative offices, magically reforming two brand new nuclear envelopes around the separated DNA. Section 5, Cytokinesis, the final split. Okay, so we've divided the genetic information perfectly, but now we have to actually cut the factory building itself in half to create two independent cells. And nature solves this mechanical problem differently depending on the organism. Animal cells use a flexible cleavage furrow to literally pinch the cytoplasm inward from the outside until it snaps in two. Plant cells, on the other hand, have rigid walls and can't pinch, so instead they grow a rigid cell plate outward from the inside. Section 6, Why Mitosis Matters. So our parent cell has been successfully reborn as two identical daughter cells. But why does this microscopic dance actually matter to you in your daily life? Well, it's the fundamental mechanism that drives the physical growth of multicellular organisms, transforming a single-cell zygote into an adult. It restores the vital nucleocytocosmic ratio, and, bringing it all back to where we started today, This exact cloning mechanism is what repairs your damaged tissues and heals that pesky paper cut. And honestly, it's absolutely mind-blowing to think about the sheer speed of this process. Get this, the cells lining your gut completely replace themselves via mitosis twice a week. No way, right? Your body is running this highly regulated, perfect cellular assembly line so fast that you get a totally new gut lining every few days just to keep you healthy. But all of this perfection begs a really crucial question. What happens when this perfectly orchestrated process breaks down? When those strict metaphase checkpoints fail and unregulated cell division spirals into cancer. I'm going to leave you with that provocative thought about genomic catastrophes. I hope this explainer illuminated the incredible power of your own biology and left you curious to discover even more about the microscopic universe inside you.